Welcome, everybody. I'm so glad to have you join again for one of our really wonderful expert talks where we really bring the top researchers who are doing some of the most important work in ALS to you directly so that you can answer your questions. You can get answers to your questions directly from them. And this week, we're really proud to have Dr. Arens Taga. His presentation, Building the Motor Pathway in Vitro to Understand ALS, it really encapsulates a lot of the work he's doing as assistant professor of neurology and neuromuscular division in the Department of Neurology at the Johns Hopkins University School of Medicine. He is a recipient of the 2025 Next Generation Research Grant from the American Brain Foundation. And Dr. Taga is a physician scientist specializing in neurology and neuromuscular medicine at Johns Hopkins. He earned his medical degree from the Università degli Studi di Parma before completing neurology residency at Johns Hopkins Hospital in 2024, and is a neuromuscular medicine fellowship and has a neuromuscular medicine fellowship in 2025. And Dr. Taga's research focuses on the cellular and molecular mechanisms of axon regeneration in ALS, with particular emphasis on the role of the beta-1 important protein in restoring motor neuron connectivity. His work has garnered a 2025 Next Generation Research Grant, and he also is fluent in Albanian, Italian, and English, and maintains a robust publication record spanning basic neuroscience, clinical neurophysiology, and neurodegenerative disease modeling. It's our pleasure to have him today to present some updates on his work. And Dr. Taga, before I turn it over to you, I just want to remind everybody, you can ask your questions anytime in the chat. We will have the presentation go in full first, and then we'll open it up to questions and answers at the end of that. So thanks, everybody. Welcome, Dr. Taga.
Thank you. Thank you so much for the invitation. This is really a unique opportunity for me to talk to this audience. Some of the patients I see in clinic weekly, patients with ALS and their family members. So it's very important to explain what we do in the lab when we are not in clinic. And so what we do in the lab and what I do specifically is in the last few years has been to find better models that can be used to model the pathophysiology, the mechanisms of ALS. And specifically, my focus has been on modeling that network, which is where ALS occurs, which is the motor pathway. And let me see. Okay, so what I'm going to talk today is about the motor pathway and how we model that using a very peculiar tool, which you have heard probably in the past, that I will explain briefly, which are these human-induced pluripotent stem cells. And then how we have been able to, by using them, to generate kind of the single elements that compose and make the motor pathway. And then kind of as a final conclusion of this work is how we have been using these models to model specific mechanisms of ALS, in particular, as it was mentioned, the role of axonal pathology and axonal regeneration in ALS, which is my latest field of research. So when I was saying that ALS is a disease that affects the motor pathway, what you have to imagine is this circuit here on the right side. ALS is a disease that affects the upper motor neuron in the motor cortex, which is connected through what we call the corticospinal tract. So this very long projecting axons, wires, to the lower motor neuron, which is the neurons that are located in the spinal cord. Those neurons are then connected to the muscle through a peripheral motor axon, and this connection occurs through a neuromuscular junction. So ALS occurs, and this is very unique of this disease. This is what defines it clinically, pathologically. It occurs within this relatively simple circuit. And there are hypotheses of where the disease starts. There are some of us that think that the disease starts at the upper motor neuron and then it propagates. This is kind of this dying forward hypothesis. It propagates downward from there using this corticospinal tract to the spinal cord. Other think that the disease starts in the spinal cord and then it propagates backwards, and we call that dying back hypothesis, or likely a combination of the two. Now, the other feature of ALS, besides this propagation from the upper and lower motor neurons, is that ALS propagates locally within the circuits that compose this motor pathway. So, for example, a patient that starts, let's say, with lower motor neurons, so spinal cord disease at the hand, the right hand, for example, it spreads from there to the more proximal, so the arm, and then to the other side, so to the left arm, and then to the legs. So there's clearly also a local propagation of the disease at the circuit level within the spinal cord and the motor cortex. And the models that we have been using to, or the cellular models that we have been using to replicate this in vitro are, as I mentioned, human-induced pluripotent stem cell. As you may know, these are cells that we transdifferentiate using mature cell source, historically skin cells or peripheral blood cells, but more recently other cell type, for example, urinary cells that you can find in samples from urine. We differentiate them using some transcription factors and generate the stem cells, so cells that can be replicated in any kind of cells of the body, including neural cells, so neurons and astrocytes. And why is that relevant? That's relevant because these cells are human, which is relevant by itself, and they have the potential to carry the abnormalities of patients with ALS. And that can be either with sporadic, so patients that do not have a genetic cause, or patients that have a familial and a genetic cause of ALS. And we can use them to make either motor neurons or astrocytes, so the glial cells that are around and support the function of motor neurons. So this is a form of precision medicine, if you want. And then we can use these cells to study and to model ALS in different ways. So we can model the pathological abnormalities. We can model the circuits and kind of the complexity of the cellular population that makes the cortex and the motor cortex and the spinal cord. We can use them to screen for drugs. We can use them to screen for genetic mechanisms or to correct genes in familial forms. And then we can use them to do functional measures through electrophysiology and other kind of functional measures, which are, for example, axonal, wiring and connection through axons. So using human models is relevant, as we all know in the field that ALS is a disease where many clinical trials have failed over time, despite trying, right? And one of the reasons for this, beside the mechanism of the disease being complex by itself, is that the models that we have used in the past have been mainly mouse models. And this also explains why the NIH has put a kind of among the goals for the future, using human relevant models for preclinical studies before going to clinical trials to decrease the number of failed clinical trials. And this is true also for ALS. So now let's go back to this motor pathway. I introduced some of the tools, and so I'm going to now talk about how we have been using this tool to model specifically. I mentioned the two regions where the disease may start, so the cortex and the spinal cord, to make in vitro the motor cortex and the motor spinal cord, which is the anterior horn of the spinal cord, so the upper or the lower motor neuron. And so to be able to do that, we have utilized some differentiation protocols that have the ability, and we have adapted them, to make regionally specific neurons and regionally specific astrocytes. So this regional specificity, in this case for the spinal cord, can be demonstrated using a number of markers that define the regional, the anatomical identity of neurons and astrocytes. For example, here you see this chat and HOXB4 for respectively neurons and astrocyte. And this can be done also for the cortex, where other markers are the CTIP2 and this OTX2, so markers of regional identity. Now, what happens is that we need both neurons and astrocyte because the cellular population of the motor cortex and the spinal cord is not just made of neurons. There's an incredibly large amount of glial cells or astrocytes, which are the supportive cells. In the cortex, the ratio between neurons and astrocytes is 1 to 1. There are around 85 billion glial cells in the cortex. And in the spinal cord, the ratio is probably even higher. So there are more glial cells than neurons, probably a ratio of 2 to 1. So we need both cell types to model this regional population of cells. And what we can do by co-culturing neurons and astrocytes is to demonstrate how neurons and astrocytes actually are talking to each other. There's a crosstalk. And how we can demonstrate that? We can show here using, again, iPS-derived neurons and astrocytes, right? So in vitro, we can demonstrate that astrocytes promote the maturation of neurons and specifically promote the connection between neurons through synapses. Here on the top, you see that when you have co-culture, so this motor neuron plus astrocyte, in the presence of the astrocyte, neurons have more receptors. So glutamate receptor, for example, here, GABA receptor and glycine receptor, which are the main receptors through which neurons communicate in the motor pathway. And the same is true also for more kind of morphological markers of synapse formation, which you see here, this synaptophysin and PSD95. So we can replicate that in vitro. We can also replicate in vitro that astrocytes promote the ability of neurons to create networks, so to connect between each other. And you can see here, again, when you have motor neurons with astrocytes, over time, this is a phenomenon that is dynamic. Look at how much more complex is the network of neurons compared in co-culture with astrocytes compared when they are alone. Other features of these connections are even more evident, like, for example, the number of neurites, which is the individual branches, how big these astrocytes are, how large their connections are in between each other. And being a crosstalk, it's true also the opposite, which is that the neurons promote the maturation of astrocytes. And this maturation is both morphological and functional. And again, here is shown that when in co-culture, astrocytes have increased expression of a number of markers, for example, S100 beta or connexin 43 or this EAT2, which are markers of morphological maturation and function of astrocytes. And finally, using this in vitro co-cultures, we're able to demonstrate how in this regionally defined environments, there's an electrophysiological maturation of neurons and how astrocytes are relevant to promote this electrophysiological maturation. And to do that, what we do is that we co-culture neurons and astrocytes in these special plates that you see here. This is an example with the one well. There are, for example, in B, also with the 24 wells, so individual culture surfaces, where under the surface where we culture cells, we have electrodes, which you see here in this box, very similar to what an EEG will look like, an electroencephalogram. So they record the electrical activity that is occurring above. Why that is important? Because through these electrodes, we are recording actually the activity of population of cells. So we are recording activity of these cells in groups and how they are connected. And what we see when we put the cells and we culture them on this kind of electrodes is that they have this electrophysiological activity in terms of spiking, which are firing of individual or few neurons, bursting, which are groups of neurons that fire together, sometimes in a very synchronized way, meaning that they are connected through complex networks. So, and here you see what the electrophysiological activity looks like when we record it on these plates, these MEA plates, through this scatter plot of electrical activity. And you see again, which is what I was mentioning before, that astrocytes, when they are present in co-culture with neurons, promote this huge increase in activity of neurons. For example, the firing rate is increased or the number of bars is increased. And of course, this is a phenomenon that occurs dynamically over time. Okay, so now, before we go to the next, so I hope with this to show you that we are able, using iPSCs to generate neurons, we are able to use iPSCs to make astrocytes that are regionally specific. So they have features of the motor cortex and the spinal cortex. We are able to plate them together, and plating them together recreate some of the normal physiological features of the motor cortex and the spinal cord. Now, we have used these models, and I'll do this back and forth also in the next slides, to model ALS. And what features specifically of ALS we have been trying to understand in the lab? One feature is the role of these cells that I mentioned, these astrocytes, these glial cells, in disease progression. And why that is important. One feature that I mentioned is that astrocytes are one of the most abundant cells in the brain and in the spinal cord. So and there's ongoing research on that, but also established research that shows that astrocytes are not just bystanders in ALS. And the other feature is that these astrocytes are actually connected in a way that is a little different from neurons. So not through axons or synapses, but through these gap junctions, which are these channels that are composed of two hemichannels, which have a pore hole in the center, which make astrocytes functionally a network that is connected throughout the central nervous system, where there's a passage of molecules between cells. And a focus of my lab has been in identifying the potential role in disease progression for this connexin 43. Connexin 43 is the protein that makes these channels and the hemichannels, and is the most abundant of the connexins in the brain. And what we found is that when we measure the levels of connexins in the brain and spinal cord of patients that have donated that had ALS and have donated their brains for research, it's actually increased compared to control. And that is true also when we measure the connexin in the CSF. Now, interestingly, when we make iPSCs from patients with ALS using iPSCs, right? What we see is that these connexin 43 levels are increased in iPSCs. So iPSCs truly maintain some of the features of the disease, including this connexin 43 overexpression. And what we have found in the lab is that when we co-culture neurons with astrocytes from patients with ALS, these astrocytes are neurotoxic. They cause neurodegeneration, the death of neurons. And we can actually rescue this neurodegeneration using small molecules which block this connexin 43, including molecules that have been tested in clinical trials for other indications, specifically migraine, for example, meaning that this medication have the potential actually to be translated relatively rapidly, which is what we have been trying to do, we'll try to do in the future, to clinical trials for ALS. Now, another feature of ALS that I just showed you before is this functional abnormalities that occur in the cortex on the spinal cord. A functional abnormality is this hyperexcitability. So at the very early stages of the disease, there's some evidence that neurons are firing more than necessary before they undergo death, neurodegeneration. And what we found is that by blocking this connexin 43 that act on astrocyte, we can actually block this at a cellular level in vitro, this hyperexcitability. So another proof that this blockade has potential for ALS, for translation to ALS. Now let's go back to the motor pathway. And so I mentioned that the disease starts either in the motor cortex or the spinal cord. It's clear, though, that regardless of where the disease starts, it spreads using this connection, this very long wire that connects the motor cortex to the spinal cord. And that wire is the corticospinal tract. What we have been doing in the lab is to model the corticospinal tract and hoping that, similar to what we did for the motor cortex and the spinal cord, to translate this model for disease research for modeling ALS. And how we have been doing this modeling of the corticospinal tract. So in this case, we have used another system, which are these microfluidic devices, it's chips. So you have to imagine this in A. This is a coin-sized plate that has two compartments. One compartment, which is where we played cortical neurons and cortical astrocytes, and this other compartment down here, which is the one where we played the spinal cord population, neurons and astrocytes. When allowed to connect, they will make these long wires between the cortex or the cortical compartment, the spinal compartment, which model the connection that I showed you in vivo in humans of the corticospinal tract. And we can do more. We can plate this microfluidic device on top of those multi-electrode array plates that I showed you, which allow for recordings, for electrophysiological recordings. So when we look at how this cell look in vitro, you see that they are here in the cortex. You have cortical neurons connecting through these long wires, this long axon, to the spinal compartment where you have the spinal neurons. And the same kind of stainings that we did before that I showed for defining their original specification, so cortical or spinal, we can replicate that in this system where they are now connected. And what we see is that the same kind of morphological and functional maturation that happens when these populations are alone can be replicated when they are connected through the corticospinal tract. And you see here in E visually how there's a maturation over time in terms of number of spikes and bursts that occur, which are these individual dots. So this complexity of electrophysiological activity increases over time and increases in both the cortex and the spinal cord. And we can measure this over time. And interesting enough, when we kind of mix and match this population, we can see that when they are arranged in a way that replicates the corticospinal tract, they achieve the highest level of activity. So what resembles the most the in vivo organization of the corticospinal tract is what works best in vitro to achieve better activity. And this population of cortical and spinal neurons with astrocytes are not just there, they are actually connected. They are connected in a way that resembles the corticospinal tract in vitro. And we can demonstrate that through some experiments. Here, we use this strategy, which is by transinfecting the spinal cord compartment with the rabies. So the rabies is a virus that can be actually, that has the ability to infect neurons and then to be translocated from one neuron to the other through synapses. And we can see that, of course, this is a rabies virus that has been inactivated. We can stain kind of the protein that this rabies virus is carrying and see that actually we have a translocation of that stain from the spinal compartment, which was transfected, to the cortical compartment that was not, meaning that there was a direct passage of this rabies protein from one compartment to the other. So those cells are actually functionally and morphologically connected. Now, to further demonstrate that they are functionally connected, we use this optogenetic strategy, which you may have seen in the past. So it's a way where you stimulate neurons and they have been made to express some proteins that are responsive to light. So in this case, it's a blue light. The neurons that are transfected and that are responsive to blue light, in response to blue light stimulation, will get active. What we see here in this graph, you can see that by stimulating with blue light, we have an initial activity in the cortical compartment in red, which is followed later by activity in the spinal compartment. Or here in this video, you see that activity on the top is followed by activity on the bottom, which means again that the corticospinal tract is active from cortex to spinal cord as it happens in vivo, in normal conditions. And here in this other kind of experimental paradigm, we did the same thing. So optogenetic stimulation, this time using glutamate blockers to demonstrate that actually this transmission of activity from the cortex to the spinal cord is mediated by the receptor for glutamate. And if we treat the cells with these glutamate blockers in here on the right side, you see that there's no more activity in the spinal cord. It goes all the way down. So they are functionally and morphologically connected through glutamate transmission. So how we hope to use, and this is ongoing work in my lab, how we hope to use this model. So we hope to use that to demonstrate that spread of the disease in ALS that occurs from the cortical compartment to the spinal compartment or the opposite using the corticospinal tract. So for example, in this paradigm, and just to show an example of how this may work, we plate in the cortical compartment neurons that carry mutations like C9orf that cause ALS. We can see some morphological and functional abnormalities in those neurons. The question is, do neurons that are healthy in the spinal cord compartment develop over time because of this connection, the same abnormalities? And the system to test for that is what I just showed you with this kind of corticospinal tract in a chip. And finally, and this is kind of the last 5 minutes, is my most recent work on, again, the motor pathway and how we translate from disease modeling, so modeling of the pathway in vitro, and we translate that to investigate ALS. So now the last part of the motor pathway. So we spoke about the motor cortex, we spoke about the spinal cord and their connection. Now let's focus on the wire that goes from the spinal cord to the muscle. So that we call the peripheral motor axon. So that is relevant because there are some hypotheses that say that the disease in ALS may not start at all in the neurons, either the motor cortex neurons, so the upper motor neuron, the lower motor neuron, but it may start in the wires, in the axon themselves, and from there go to the cell. And this is called Dieback hypothesis, and which is relevant by itself as a disease mechanism. But as we translate from disease mechanism to treatment of ALS, looking at the wires and the peripheral motor axons is relevant also in terms of therapeutics. It's clear that even if we find a strategy that protects neurons from neurodegeneration, from death, we need strategies to promote those neurons that survive to be able to take over, to reinnervate the muscles that were left de-innervated from a neuronal degeneration. So we need to promote reinnervation. Studies on tofersen that are ongoing have actually shown that the surviving neurons, this one here on the right side, are able to do so if they are left able to survive, right, through gene therapies in this case, and they do so through a collateral reinnervation. So it's clear that beside neuroprotection, we need strategies to promote this collateral reinnervation, which is why my interest has moved to the peripheral motor axon. And what model I've been using to investigate that is kind of similar to the paradigm that I showed you with the microfluidic devices, which is I use, again, these devices where I can culture the cell in one compartment, the neuron in one compartment, and I allow the axons to grow and kind of attract them to go from the cell body compartment to the proximal axon and then to the distal axon. So they are kind of stretched. And here you can see on the right how this stretched axons look like. And then what I do as a model of axonal degeneration, a very specific one, which is axotomy, which means cutting the axon, so mechanically destroying and removing the axon. You can do then by vacuum aspirating the axon in this compartment. And then if you give it time, it will regrow. And so you can model axonal regeneration. So axonal degeneration and then axonal regeneration. And you can see it here, how it looks like when we look under the microscope. So healthy axons, and then vacuum aspiration, they disappear. And then here you start seeing them reappearing. So this model is that I've been kind of translating from my prior studies. I've used to investigate a very specific new mechanism of axonal regeneration that could be relevant in normal conditions, and I hope also in ALS. So my hypothesis, and this is the normal condition situation, my hypothesis is that this protein, which is called beta-1 importin, which we know from prior studies is involved here, you see in one, is involved in the nucleocytoplasmic transport. So it's a protein that we know is present at the membrane of the nucleus, and it allows proteins to be translocated from the cytoplasm, so from the outside of the nucleus to inside of the nucleus, where these proteins then are important for gene expression. And a model that I have developed and I'm trying to confirm in control condition is this. So beta-1 importin is not just expressed at the nucleus level, but is also expressed in the axon. And specifically, beta-1 importin is expressed in the axon after axonal damage. And what I hypothesize it happens is that this beta-1 importin is translated locally in the axon. It binds to protein in the axon that are important for axonal regeneration. It allows their transport from the peripheral axon to the nucleus, where it does the job that I just mentioned, so the translocation from the outside of the nucleus to inside of the nucleus. And this results in a number of changes in gene expression that then promote axonal regeneration. And my preliminary work, which is work sponsored by the AAN and the Brain Foundation, has been in showing how actually you see here in B, when you have an intact axon, beta-1 importin is expressed mainly in the nucleus, and the expression in the axon is very minimal. So the stain here is really faint, very minimal. And in C here, you see kind of the same, right? So this pink color is to stain the axons. And you see that the beta-1 importin, which is in green, is really not at all or minimally expressed in the axon that are labeled by this pink color. But when I do the axotomy, so when I cut the wire, which is here, you see how this green lights up, meaning that there's an increase of expression of the importin in the axon. And we can demonstrate that also with Western blot, which is a way to quantify proteins. And again, I do the axotomy here, and then I allow axonal regeneration. And when I measure protein levels of importin at this level, so just proximal to where the axon has been cut, I see that there's an increase in the importin beta-1. And if I prevent the expression of importin beta-1 through this strategy, which is called RNA silencing, which is this plus here, you see that the protein does not go up. And this is relevant for axonal regeneration because when we look at the axonal regeneration on this side, when I use the silencing strategy, so I prevent importin from being expressed in the axon, there's not much axonal regeneration. When I do not use siRNA, so I allow importin to be expressed, there's this axonal regeneration. And so why this is relevant in ALS, beside the fact that this is relevant itself as a strategy to promote axonal regeneration, so the rewiring and the reinnervation from neurons, there is also evidence that in ALS, axonal regeneration is disrupted. Not only neurons in ALS undergo neurodegeneration, but damaged axons are less able to repair themselves. And this has been shown by many groups. Here, as a proof of concept, as this is relevant to the platform that I'm going to use, I show that just by using the extracellular media, so the solutions that we use to keep these neurons alive and going, from neurons that carry the C9orf mutation, so the most common familial form of ALS, I can cause an axonal degeneration. So that media alone is sufficient to cause damage to the axon. And my hypothesis here, which is kind of the equivalent, but now in ALS to what I showed before, is that the reason why this may happen, this inability of axons to regenerate in ALS, is because this beta-1 importin may get trapped in the cytoplasm, so outside of the nucleus, in protein inclusions that are a pathological feature of ALS. So in the pathway that I showed you before, when I induce an axotomy as a model of axonal damage, I hypothesize that importin is expressed locally as it should, but then when it's transported towards the nucleus, what happens is it gets trapped in these protein inclusions. So it's unable to carry its work on promoting axonal regeneration, which is deficient in this model. So I think with this, I kind of reviewed the whole kind of story about disease, about modeling the motor pathway in normal conditions, and how by going from that kind of basic understanding, we can translate that and ask very specific questions about disease mechanisms in ALS. And much of this work was done during my time as a postdoc in Nicholas Maragakis' lab, including some work done by Ariana, who's a postdoc in his lab that did some of the study on the corticospinal tract. And this work has been sponsored by a number of grants, which I will say that are decisive for me as I start my independent career as a PI. I actually just started my lab, I was saying a week ago. In a time of difficult funding, these associations and the larger community of ALS has really been supportive and decisive for my work. And I think with this, I will stop for questions.
Thank you so much, Dr. Taga. Really great information, really in-depth look into what's possible and how fast it's moving now, much faster than in the past, from bench to potentially bedside with all of the new precision medicine approaches that are being taken. And thank you for supporting that kind of work. In ALS, we're looking and working for curative and disease modifying therapies for people living with ALS today. So we're really passionate about the kind of work that you're doing to support that. We do have some questions from the audience, so I'm going to kick it off with the first one being that for someone who's been living with ALS for 6 years and still retains most movement, what kinds of clinical trials would they be eligible for? Are there better ways, and along in those same lines, are there better ways to match patients like this to relevant trials?
Yeah. So, I mean, that's a very important question in the field that has to do much of how we recruit patients in clinical trials, right? And let's do one step back. So a problem is many of the patients know, and the doctors have learned from patients, is that the ALS is so variable clinically, and such that, I mean, many of us may think that those are actually different kind of diseases, though they kind of converge to a similar final kind of manifestation. And when recruiting patients for clinical trials, so a problem to face is this huge clinical variability. And among the clinical variables, which is the most important for patients, clearly is how fast disease is progressing. So what has happened in the past is that there has been recruitment that was not as well thought, including patients at different stages of the disease, which has caused for many of these clinical trials to actually fail, because probably they were not capturing meaningful clinical changes as the patients were so different. That has caused many of the clinical trials to kind of be very strict with criteria, and that has caused problems with let's say two extremes of the spectrum of the disease, which are patients that are either very advanced or very fast progressing, or patients that are very slowly progressing. And so a way to overcome that is to build better clinical trials. And so I understand the fact that it's very frustrating for many of our patients that are in these extremes, right, to be enrolled in clinical trials. And that is because the way we have built clinical trials has been deficient until now. And the field, though, has moved, and much of the work that is being done at MGH currently is to try to make better clinical trials, including this kind of phenotypes at the extreme opposites. So we don't have now good strategies for this kind of patients, and that is an unanswered need in the field.
Thank you.
All right, the next question is, do we have iPSCs for propiospinal neurons?
That's a great question. So in general, sensory neurons and non-motor neurons are classes of neurons that we can make from iPSCs. They are much more difficult to generate in vitro. There's some evidence that there's propriospinal dysfunction and network disease in ALS. We don't have this ability to kind of pinpoint this other cellular populations. The field has tried different approaches, for example, to make oculomotor neurons as examples of other neuronal populations that may be not affected or affected at later stages, or that are kind of behaving differently from motor neurons, to understand why they do behave like that. The tools, unfortunately, are still limited in making this kind of this other type of neurons, but it's it's a very potent strategy to investigate also the question of why these other neuronal populations are so resistant. There are hypotheses. We can talk about that, but one hypothesis, for example, is that other classes of neurons have more calcium binding proteins, for example. Another hypothesis is that motor neurons have direct connection to, particularly the spinal neuron, has a direct connection to the sensory afferent from the muscle, from the spindle cell kind of system in the muscle, and that can make this motor neurons particularly prone to neurodegeneration. So those are important questions. One way to address that the field is kind of moving is to use in vivo, kind of autopsy or tissue or cellular cultures where we do spatial transcriptomics, because then we can separate the individual cell types and then investigate and ask this question about what makes some neurons more affected and others less. But I would love to be able to get those kind of neurons in vitro. It's very difficult.
Got it. Thank you.
Next question is, if the ALS is slow progression, are there likely chances of improving the neuron communication? Is it more possible that slow progressors might have that chance of improving the neuron communication?
Yeah, so that's absolutely true. So what happens in ALS, there are two kind of phenomena that happen in parallel. And one is neuronal degeneration, so neuronal death. The other one is that the surviving neurons are, meanwhile, while neuronal degeneration is happening, are reinnervating. So what causes disease to progress is the imbalance between the ability of the surviving neurons to reinnervate and so cope with the loss of nerve muscle communications, which is what's causing weakness and neuronal degeneration. So of course, those patients that have a slow pace of neurodegeneration, they have more time to reinnervate, which by itself probably explains why these patients go a little slower. So these patients are those that are ideally the best candidate for strategies of treatment, where our goal is to promote the ability of the surviving neurons to make collateral sprouting, and so collateral reinnervation of the surviving muscle. And just to add into that, the strategies that we hope to use, which are these neuroprotective strategies, meaning are targeting directly the survival of neurons. What we hope to do with that is actually to keep them sufficiently and long enough for the surviving neurons to reinnervate. So kind of to recapitulate this, which is what is observed in slow progressive patients.
Thank you so much. Next question is, does darafenacin, which we've heard about in a prior talk recently, does it also work to block the CX43 activity? As you mentioned, some migraine medication does, or does it use some other mechanism if it does?
Yeah, tofersen.
I think you're correct. The question was originally darafenacin, but
yeah. So I don't know specifically about the effect on connexin 43. of that molecule, but I can tell you about this specific molecule, which is tonabersat. So tonabersat, I think this is an interesting story that Dr. Maragakis and me in the lab, we have been working on for the last 5 years. So tonabersat is it's a small molecule that was developed maybe 15 years ago for migraine with aura. And it was actually tested in a clinical trial on patients with migraine with aura. So it has passed all the stages of clinical trials. And what it does, it is a relatively specific blocker of the connexin expressed in astrocytes in the brain. It does not affect other connexin. Connexins are widespread molecules. It does not affect other types of connexin in other organs, making this quite safe. In fact, it was a very safe medication in this trial. We have actually used that same molecule in vitro, and we have shown that by blocking connexin 43 in this cellular model, we prevent the astrocyte-mediated neuronal degeneration. Not only the neuronal degeneration, but some of the functional features that are early on in the disease, which are, for example, I said this hyper-excitability, this increased activity of neurons that maybe precedes their death. And so tonabersat is a medication or similar medication to tonabersat that we hope to be able to translate now in a clinical trial. There have been some administrative challenges, but I hope that will be something to use in the future. And I have to say that it's also one of the reasons I decided after finishing my postdoc to continue to stay here. It was the dream, right? To see something that you have tested in a dish, that you know that is safe, that has been tried for other conditions, and to translate that to ALS. More downstream to this kind of mechanisms, I showed you gap junctions are these channels that connect astrocytes, not only, but in the CNS astrocytes. They can be facing each other and make so a hole, a pore between the cells, or they can be just on the membrane, so the external side of the cell, and exposed to the outside. When they are exposed to the outside, what they do is that they release maybe toxic compounds from the cell outside. And these toxic compounds are what we think responsible for astrocyte-mediated neurodegeneration and neuronal death. One of the molecules that is released that we have shown in the lab is ATP. And ATP may cause neuronal death by binding a specific receptor on neurons. So another way to tackle this pathway is to go more downstream to block ATP binding to these receptors, and which is something that is feasible, and we have published on that because there are classes of purinergic, so ATP receptors that can be used and are on advanced stages for disease treatments. They have been tested in other diseases like depression, for example, in terms of nervous system conditions, and also retinal diseases. And retina is an extension of the brain at the end. So that is another approach to the same pathway. Thank you.
Right, the next question is, are you aware of any home therapies or remedies that patients can take that would promote neuron regeneration?
Yeah. So, this is kind of the classes of vitamins or amino acids or things like that. There's actually a website where you can see a list of this kind of. Now I cannot remember. I'll just check in a second and put that in the chat. But there's a website where experts have kind of made some comments about which of these treatments or approaches can work. Unfortunately, in general, in neurology, there's not evidence that if, for example, a vitamin level is normal, by giving more of that vitamin, that is going to cause more effects from the vitamin. In general, they work if the vitamin is deficient. And so in general, there has been not much success in trying these approaches, right, in neurology and in ALS. There are, though, some attempts. For example, high doses of B12 is an approach that many patients do, and some doctors in the field also suggest, which clearly is not toxic. And so when my patients ask about, this kind of approaches, I will say that B12 is okay, safe, and many patients use it, and that's an okay approach in my opinion. But other approaches may be something to take with more caution, because we know that other vitamins, like for example B6, when in a very high level, actually they can cause damage to the nerves. Some caution with some of this, but other, like B12, for example, that's a common approach that many experts in the field use, and I think it's safe.
Thank you. The next question is both a comment and question. Last year, the University of Copenhagen and University of Rochester showed that healthy glial cells implanted in the brain totally kind of outcompeted diseased or dying glial cells. Could that process assist in repairing the diseased neurons?
So that's a great question, and a great scientific approach. And at Hopkins, there has been a long history of research about using healthy glial cells, right, and implanting them in animal models as a mechanism to kind of help with and prevent either disease spread or neuroprotect neurons. The opposite being that many experiments have been done, including some of the first one done here by Dr. Ralston and then Dr. Maragakis too, which was to use astrocytes from patients with ALS or mouse astrocytes that carry SOD1, implant them in healthy mouse. And what happens is that actually there's neurodegeneration. So to say that there's some rationale behind that. Now, the problem with doing this currently, but is something that there's ongoing work on, is that astrocytes derived from iPSCs are not yet at the stage where they are safe enough for transplantation in the majority of cases. The field, though, is going towards making these iPS-derived neurons or astrocytes stable enough that when they are implanted in human brains or human spinal cord, they maintain their differentiation, they maintain their mature phenotype, and so they're safe. Because what may happen, and we don't want that, is that they actually start to de-differentiate, to replicate and make a tumor. So that's why there has been some hesitancy of using this iPS-derived astrocytes or neurons for an implant, or also as a way to kind of substitute, to take over the dying neurons, or as you were saying, as a mechanism with astrocytes to neuroprotect neurons. So, but yes, that is absolutely a way to go. And there are other type of astrocytes that are derived from embryonal stem cells, so from embryos, that may be safer, and there are actually some that have been clinically validated, which is what you may see for Parkinson's disease. There are some very recent publications that use this approach to make dopaminergic neurons and to transplant them and to show actually some benefits. Those are embryonic derived neurons, dopaminergic neurons, which adds some, ethical questions, and also, of course, the fact that that's not a source that is so available, that is so, widespread. So it's a very limited source.
Thank you.
So next question is, what is the timeline that you envision for your research to start translating into potential results or potential therapies for patients?
So for now, so two separate kind of directions. So the connexin story is something that we are actively actually looking for starting a clinical trial. And so that is something, I mean, hopefully there has been some administrative difficulties, but in the next 1 or 2 years. Now, the second part, which is the axonal regeneration. So we are really at the early stages of understanding this mechanism in vitro. And so that is something that is a little difficult for me to determine like with accuracy. But the tonabrestat story and the connexin, yes, it's something that is much more close to clinical trial.
Got it. Thank you.
Thank you. And so the next question I have for you is, I've read that exercise, like running and biking, can incite neurogeneration. If only we could, do you agree? If we could do that.
Yeah. So what the exercise does is that when muscle is active, releases a kind of a hormone, which is called myostatin, among others. This hormone actually, we know, promotes directly axonal regeneration. And that is the basis of us in the field when we see a patient that has a nerve damage, to suggest that exercise is a way to promote the recovery of the nerves in general, but also of the motor wires and the motor axons. And so, yes, absolutely. Exercise is a mechanism that promotes axonal regeneration. Now, in ALS, there are many studies that have looked at that. And there's evidence that patients that do some aerobic exercise, and particularly patients that do stretching exercises, that has some benefits in terms of, beside quality of life, in terms of disease, in disease progression, and also helps a lot in preventing contractures, which is a non-neurological cause of weakness in many of the patients, right? So we want to prevent that from happening. Strategies to use some of the effects of exercise, like this myostatin expression, for example, as a way to promote nerve regeneration and axonal regeneration, have actually been used. And myostatin has been kind of targeted by some trials years ago. It did not work, but that does not mean that a better version of activating that pathway, which is the normal one, so real exercise, is the best way to go for now, until we understand that better.
Thank you.
All right, the next question is, can the iPSCs be upscaled into organoids?
Yeah, so what I showed you is two-dimensional cultures, right? So we play the cells on a surface, and this strategy that we have used is the reason we use these two-dimensional cultures is mainly because we can have a direct control on the cell composition. We can decide what kind of cells are there. We can combine these cells the way we want, and we can arrange them in vitro the way we want. So it allows a lot of manipulation. Now, another way to go is to allow the cells to make, particularly the cells at the earlier stage of differentiation, when they are, we call them neural progenitors. When you allow the cells to float in a medium, in an extracellular solution, what they do, thanks to intrinsic cellular programs, genetic programs, they will start to differentiate and make these three-dimensional structures, which resemble actually the central nervous system. And they will start to arrange in layers in a way that, depending on how we manipulate the media, resembles a little the brain. So we call this kind of mini brains. You can make the same with the spinal cord, and by manipulating kind of this intrinsic cellular program through growth factors or patterning molecules. And so you can make kind of spinal cords. And actually a group has made what I showed you, this kind of corticospinal tract connection using this two population of organoids, cortical organoid kind of resembling the brain and the spinal one. And those are tools that are very interesting for sure for development, because they allow us to understand how the brain and the spinal cord develop. The main problem there is that we don't have much control on the cellular composition of the structures. So we cannot interrogate specific questions at the cellular level, because we don't have this ability to manipulate cells individually, as we can do when they are at two-dimensional cultures. But the field, and this is something that is ongoing, is trying to use organoids to model pathways and networks similar to what I showed you for the corticospinal tract, and similar system have been used for the sensory pathway, for example, or for the dopaminergic pathway system, more relevant to Parkinson. So that's kind of, but we do organoid cultures in the lab, and it's just that for this experiments we have preferred something more flexible.
I'll jump into the next one, Dr. Taga. Given the movement towards precision medicines and the urgency to get there, do you think there's a possibility that this technology the modeling technology you're developing can replace the animal model stage of getting to trials with these precision interventions?
Yeah. So yes, the answer is yes. The field of ALS, I think, very clearly is now using iPSCs for testing medications. And of course, animal studies remain important, particularly when we ask questions about drug delivery, target engagement, dosing. So for the drug development kind of programs, they remain very important, and I mean that's a way to do that safely when we translate to humans. But when we go to the very basic drug discovery part, we have been consistently now using iPSCs, I would say, in the last few years, and that is one reason for that. In this, yes, the precision medicine is also because I mean clearly the approach of with animal models in ALS has failed, and beside our lack of understanding of the mechanisms in the majority of patients, right, the sporadic patients, the non-genetic patients, is also that the models that we're using were incorrect. The rodent, the mouse model is an artificial model. Of course, mice, they don't have ALS, and we overexpress a human gene in mice. They have a very kind of peculiar disease that may have some features of ALS, and has been historically helpful to develop medications, but it has not brought us to target disease at the molecular level and with the precision we want to treat patients. So yes, this is where the field is going.
Two final comments. One is thank you so much for the work you're doing. Hopefully it can also lead to better indicators of success in a trial, like beyond neurofilament, but actually being able to track quickly and within the time window of a trial, the kind of reinnervation that we're looking for in some of these studies. So we really hope that you're a part of not only the research, but also the development and the success of bringing more therapies to market. One last question is there anything we can do to support your research, especially given the current funding environment?
So, of course, the foundation grants are what keeps ALS research going currently. And I mean, as you know, the NIH funding has important challenges right now. And I have to say that in my case, different institutions have been and foundations have been very supportive. I mean, you heard about the American Brain Foundation, and there's also a fight MND and finding a cure through MGH. So those are already foundations that have been very helpful to me. And what you do is amazing because, I mean, I was talking initially before we were offline. One of the reasons I came here to do research in the United States is the very unique opportunity to be a clinician and a scientist. And what makes this possible here is that there's this kind of support, which is unavailable, I have to say, in many other countries, including Western countries, including for me, Italy. And so what you do and kind of the attention you give to the basic scientists is really already outstanding and helpful. And for me, particularly as I just started, doing some independent work.
Thank you. Thanks for the great presentation. Thank you for all the answers you provided. Thank you to the audience as well, to our community for providing some really good questions to open up the dialogue even more. And with that, McFinn, I think we will end tonight's segment of the expert talk and turn it back to you in the community for some discussion.
Okay. Well, one thing I'd like to ask Dr. Taga,
Yes.
how did you like the audience that you had tonight listening to you? Did you find the questions good? And
yeah, the questions, and I have to say that patients with ALS, they know their disease very well. They are very well informed. And this is an experience that I find in clinic, and I found here also. And the scientific level is actually very high, right? I mean, very spot on questions that the scientists will be asking, like organoids and astrocyte transplants. Those are actually our tools, our conversations, and our experiments. And so clearly they have heard from the best, right?
well, you're one of the best, and we are your team. So thank you. You can always say, I really need help in this specific area, like whatever it might be. You have everything ALS and everyone connected with it. Very appreciative of you, your time, and that you came into everyone's living room tonight.
Yeah,
nobody had to go anywhere.
Yeah, I see that. I see on the videos number of patients, and I wanted just to say hello to all of them for listening to me tonight. I try to explain, to the best of my abilities. But I'm really happy that I was, it's unbelievable, right? That we're able to communicate with patients and just to be heard in their houses, and this is great. Many of them all together, it's fantastic. Thank you.